Hello everyone. In the previous video, we were discussing about the biotechnology and its applications. In that, the first we have studied about the application of biotechnology in the field of agriculture. So now let us move to the next one, that is application of biotechnology in the field of medicine. So we can call it as medicinal biotechnology. Application of biotechnology in the field of medicine. It mainly includes the production of various kinds of drugs or the medicines. It may be the antibiotic or it may be the antibodies, for example, monoclonal antibodies. The various kinds of medicines are produced by using the various types of biotechnological processes, and the development of various kinds of therapeutics or therapies. One therapy will be discussed in this chapter, that is gene therapy. So the treatment technologies are developed by or contributed by the biotechnology, as well as the diagnostic methods, the various methods to detect the diseases. So these are some of the very important applications of biotechnology in the field of medicine. Now, before moving to the application of biotechnology in the field of medicine, let me explain the structure of pro-insulin as well as insulin here. The reason is, so at the beginning we are going to discuss about the production of artificial insulin, which is used for the treatment of diabetic patients. So, but before knowing about that, we should know the structure of insulin, the importance of insulin. So already you have studied about the insulin in the previous classes. Insulin is a hormone, and that insulin is produced by the beta cells of islets of Langerhans of pancreas. And the insulin is responsible for the maintenance of constant glucose level, the optimum glucose level in the blood. So that is the importance of insulin. Just remember, the insulin is not produced as it is. It is produced in the form of pre-pro-insulin. And then the pre-pro-insulin undergoes enzymatic digestion or the breakdown that leads to the formation of pro-insulin. And the pro-insulin is then converted into active insulin. So this is the biological synthesis of insulin in our body, in the body cells. So here that explanation is not given, but just the structure of pro-insulin as well as the structure of insulin is explained with the diagram. So these two diagrams are very important for exam. So you should learn the diagram and you should know the explanation for this. Let me explain the structure of pro-insulin first. So this is the diagram of pro-insulin. As I told earlier, this is a primitive form of insulin. This is not the functional protein. So here the pro-insulin, it is made up of 3 polypeptide chain. It is a protein. It is made up of 3 polypeptide chain. In the diagram you can see one is A chain, another one is B chain, another one is C chain. So this A chain is also called as alpha chain or the acidic chain. B chain is also called as beta chain or the basic chain, and the C chain is the connecting chain. So here A chain as well as B chain are connected by the C chain. So these are the polypeptide chain. These are the 3 polypeptide chains. Okay, and here the in pro-insulin, the A chain as well as B chain are interconnected by the disulfide bond. So this is the structure of pro-insulin. I repeat, in mammals including humans, insulin is synthesized as pro-hormone. Like proenzyme, the pro-hormone also needs to be processed before it becomes a fully mature and functional hormone. So here the pro-hormone, which can contain alpha chain, beta chain, and an extra stretch called as C peptide or the C chain. So this C peptide or the C chain, connecting chain, is not present in the mature insulin and is removed during the maturation into insulin. That means the active insulin is made up of only 2 polypeptide chain. Let me explain the structure of insulin now. See, this is the diagram of active insulin. Here you can see only 2 polypeptide chains are there. One is a peptide, another one is B peptide, and these 2 polypeptide chains are interconnected by, they are linked by, or they are paired by the help of a disulfide bond. So here is the explanation. Insulin consists of two short polypeptide chains, that is chain A as well as chain B, that are linked together by the disulfide bridges. So this is regarding the structure of proinsulin as well as insulin. These two diagrams are very important for exam. Okay. Now let me explain the production of genetically engineered insulin. Genetically engineered insulin means artificial synthesis of insulin by using the genetic engineering. See, in the older days, the diabetic patients were treated by using the insulin isolated from the cattle as well as pig. But such insulin uh, that showed benefit or that helped to treat some of the patients, but in most of the cases that resulted in allergy. So in such condition, the scientists thought to produce human insulin, means which is exactly similar to the insulin synthesized by the human body. And such insulin is produced by the genetic engineering technology. You know, insulin is a protein. It is a proteinaceous hormone. So proteinaceous hormone or the protein is basically made up of amino acids. And the sequence of amino acids and the number of amino acids is decided by the codons present on mRNA. And the sequence of codons is decided by the genetic information present on gene. Gene is nothing but a short segment of DNA, and that is found in the chromosome in case of eukaryotic cells. So the genetic information present in the DNA is responsible for the production of a specific type of protein. So this is called as gene expression. This is explained in another chapter, that is molecular base of inheritance, gene expression. So in the first step, that is called as gene expression 1 or transcription. Synthesis of RNA takes place according to the information present on DNA, that is gene. And in the second step, translation. According to the information present on mRNA, the protein molecule will be formed. So mRNA contains codons. Each codon represents one specific amino acid. So the sequence of codons present on mRNA determines the sequence of amino acids in the protein. So if you know the sequence of amino acids in a protein, we can find out the sequence of codons present on mRNA. If you know the sequence of codons present on mRNA, we can produce an artificial gene coding for that particular mRNA. So by this, artificial genes were produced, and such artificial genes were used for the synthesis of artificial insulin, that is called as humulin. That is called as humulin. Let me explain. So management of adult onset diabetes is possible by taking insulin at regular time intervals. So diabetic patients are treated by providing the active insulin at regular time intervals. Insulin used for this was earlier extracted from pancreas of slaughtered cattle and pigs. So the functionally they are similar, but somewhat there is a difference in the sequence of amino acid. So insulin from an animal source, though caused some patients to develop allergy or other types of reactions to the foreign protein. So it resulted in some negative problems in the patient body. That's why they thought to produce the human insulin artificially. The main challenge for the production of insulin using recombinant DNA technique was getting insulin assembled into a mature form. So it is very difficult to produce active insulin as it is. Such technologies are also developed. There are two methods. In one method, A chain as well as B chain polypeptides are produced separately, and then they are allowed to undergo disulfide bond to produce an active insulin. In another method, pre-proinsulin genes were isolated from the human body cell, that is beta cells of islets of Langerhans of pancreas, and such proinsulin genes were processed, that means allowed to undergo transcription that results in the formation of mRNA, and then from that mRNA again the complementary DNA were produced by the reverse transcription method, and such complementary DNA were linked to the LAC-Z gene, then inserted into the vector, followed by inserting into the host cells, bacterial cells. Such bacterial cells were cultured to produce a human insulin. So this was one method. But here another method is mentioned, that is production of A chain as well as B chain separately. But the thing here, the explanation is not given, just mentioned regarding the production of artificial insulin. See, in 1983, Eli Lilly, an American company, prepared two DNA sequences corresponding to A and B chains of human insulin and introduced them in plasmids of E. coli to produce insulin chains. So this is just the idea of artificial insulin synthesis. So in 1983, Eli Lilly, an American company, particularly one scientist named Itakura et al., he gave this principle. So two DNA sequences corresponding to A and B chains of human insulin were produced. Artificial DNA sequences were produced, artificial genes were produced, and then such artificial genes were introduced into the E. coli cells by using a vector, that is PBR322. So PBR322 was used as vector to produce the artificial insulin by using the E. coli cells. So chains A and B were produced separately. These are the polypeptide chains by the expression of A chain in one bacterial cell, by the expression of B chain gene in another bacterial cell. A chain polypeptide as well as B chain polypeptides were produced separately. And such polypeptide chains were extracted and combined by creating disulfide bonds to form human insulin. So this is the explanation given in our textbook. Very simple explanation is given. So this is not the detailed explanation, but for exam, this is more than enough. Okay. But let me explain the procedure in detail just for your information. No need to remember it for the exam. Okay, just to give the idea regarding the artificial synthesis of human insulin, I am explaining this, and with the diagram I will explain it. See here, the human insulin synthesis. Human insulin, artificially synthesized human insulin, is named as humulin. This is a trade name. So this methodology was proposed by Itakura et al. In 1977. So here, artificial genes coding for A chain and B chain were produced, and such artificial A chain gene and B chain genes were inserted into the vector, that is PBR322. So the joining of A chain gene to one PBR322 and joining of B chain gene into another PBR322 resulted in the formation of recombinant DNA. Let me explain with the diagram. See here, this is the diagram, and this is the diagram of recombinant plasmid. That means this is the PBR322, and here it consists oocyte. It consists two types of antibiotic resistant genes, that is ampicillin resistant gene as well as tetracycline resistant gene, and it consists many number of cloning sites. But here, see here, the A chain gene, is inserted into the PBR322 at the region of tetracycline resistant gene. Just remember, this A chain gene is associated with the LAC-Z gene. Here, LAC-Z gene provides promoter for the expression of this particular A chain gene, because the artificially synthesized gene does not contain promoter as well as terminator. That consists only the coding sequences. So here, the LAC-Z gene provides promoter as well as terminator. So it is associated with or linked with the LAC-Z gene, then inserted into the vector, that is PBR322. So here, this is another one, the same type of vector, that is PBR322, separate vector To this vector, B chain gene is inserted here. Again, that is also inserted in the region of tetracycline resistant gene. And this B chain gene is also associated with the LAGZ gene. As I told earlier, it provides promoter as well as terminator for the expression. Now, these recombinant plasmids or recombinant DNA are introduced into the or transferred into the E. coli cells. Just remember, separately, not to the same bacterial cell, to different bacterial cells, such recombinant DNA were introduced. So this is a bacterial cell with recombinant DNA consisting A chain gene, and this is a bacterial cell transformant consisting the recombinant DNA with B chain gene. Now, such bacterial cells were cultured in the bioreactor. For the large scale production, we are using the bioreactors. This was explained in the previous chapter. So such transformants were cultured in the bioreactors. When the bacterial cells are cultured in the bioreactors, once again separate culture. The first, these bacterial cells are cultured in one bioreactor, and these bacterial cells are cultured in another bioreactor. And during the culturing of these bacterial cells, normally the lactose is provided, because when the lactose is present, there will be the expression of LAGZ gene to produce a beta galactosidase. But we know the LAGZ gene is associated with the A chain gene in one bacterial cell, B chain gene in another bacterial cell. Now, when you provide the lactose in the medium, there will be the inducing of a LAGZ gene. During the expression of LAGZ gene, there will be the expression of associated gene. So this results in the production of A chain with the beta galactosidase in one bioreactor or in one type of bioreactor with bacterial cells, and in another bioreactors, the expression of recombinant DNA in the bacterial cells results in the production of recombinant protein consisting B chain as well as beta galactosidase protein. So these proteins are the hybrid proteins. These are the recombinant proteins. So these proteins are produced separately in separate bioreactors. But we want only the desirable gene product. We need the gene product, that is A chain polypeptide from one bioreactor and B chain polypeptide from another bioreactor. So we need to remove that beta galactosidase. For that, we are using a chemical treatment that is called a cyanogen bromide treatment. So here, the cyanogen bromide treatment for the removal of beta galactosidase from A chain in one bioreactor or from one culture. So this is a chain, only A chain is removed. Say only A chain is available here. The beta galactosidase protein is removed from it. And in another case, the cyanogen bromide treatment causes the removal of beta galactosidase, and the resulting product is only the B chain. So we will get A chain from one bioreactor, B chain from another bioreactor. Now we want the active insulin. The active insulin is made up of A chain as well as B chain. So we need to link these two polypeptide chains. So here, when the A chain is linked to beta chain or the B chain, it results in the formation of insulin chain. But just remember, this is not the active insulin. The active insulin, the structure of active insulin is already I told in the previous slide. So here, active insulin is produced by the purification and mixing, and then allowing them to undergo disulfide bond. So this results in the formation of active insulin. So here, this is a A chain and this is a B chain. Between A and B chain, there is a disulfide bond. So this is the methodology of production of artificial insulin. But this is not explained in detail in our textbook. No need to worry. Just remember the previous points, what I given in the previous slides. So such points are enough for the exam. This is just for your information. If you understand this particular concept, then there will not be any problem regarding the processes of recombinant DNA technology, what we discussed in the previous chapter. So that is the general step. Here, the example for that. This is an example for the processes of recombinant DNA technology. Hope you understood. Okay, now let me move to the next topic, that is the application of biotechnology in the field of medicine. There is regarding the therapy, that is the therapeutics or the therapy, gene therapy. You know the different types of therapy, chemotherapy, radiotherapy for the cancer treatment, as well as in naturopathy, mud therapy, water therapy, phototherapy, the various types of treatment technologies are there. So here the application of biotechnology or the recombinant DNA technology in the field of medicine, and that is gene therapy. Therapy means treatment, gene therapy. So gene therapy is mainly done for the treatment of genetic disorders. There are more than 6,000 types of genetic disorders found in human beings. The genetic disorders can be classified as gene disorders as well as chromosomal disorders. Most commonly, the gene disorders, especially the homogenic, that means single gene disorders, can be treated by the help of gene therapy. You know, the genes are responsible for a specific character. It may be the morphological character or it may be the physiological character. It codes for a character. The character is expressed in the form of a product. The product is nothing but the protein. So when a gene is not functional, it causes the abnormality in the body structure or the body function because of the malfunctioning of the genes. When the gene becomes malfunctioning, it may produce malfunctioning protein, or sometimes it may not produce any proteinaceous product, no product. Both causes some problem in the body, some physiological problem in the body. So how to cure such kind of disorders? When there is no expression of a particular gene, or when there is a presence of malfunctioning gene, the resulting product becomes malfunctioning. In such case, if you provide that particular proteinaceous product to the body, it is possible to cure that particular disorder. See the term disorder. Disorder is different, disease is different. Disorder is because of imbalance. It can be cured by balancing the imbalanced component. And disease is due to infection. The disease can be cured by destroying or killing or eliminating the disease causing agent, that is a pathogen. So this is the difference between disease and disorder. Here, this is a gene disorder, genetic disorder. So gene disorder can be treated by the gene therapy. So gene therapy is a collection of methods that allows the correction of a gene defect that has been diagnosed in a child or embryo. So this is just like the correcting the problem, correction of the problem. So gene defect is corrected in this gene therapy technology. So in other words, gene therapy is nothing but the replacement of malfunctioning genes by healthy, normal, functional gene to cure the gene disorder. So this is another simple definition. So here genes are inserted into the person's cells and tissues to treat a disease. Here genes are inserted into the person's cells or tissues to treat a particular genetic disorder. Sometimes we can call it as disease. No need to worry about this. Gene disorder. The most common term is gene disorder. Sometimes we can use the term gene disease. No problem. Okay. So now the correction of genetic defect involves delivery of a normal gene into the individual or embryo to take over the function of and compensate for the non-functional gene. The intention of correction of a genetic defect is to cure the gene disorder. So how to cure that gene disorder? By replacing the malfunctioning gene by healthy normal functional gene. So here normal gene is introduced into the individual or embryo to take over the function and compensate for the non-functional gene. So instead of non-functional gene, the newly introduced healthy functional normal gene will perform its function so that it will cure the gene disorder. The first clinical gene therapy was given in 1990 to a 4-year-old girl with adenosine deaminase deficiency. So the first genetic gene therapy was given to a child suffering from suffer from a genetic disorder known as adenosine deaminase deficiency, and this disorder is specifically known as SCID, severe combined immunodeficiency syndrome. SCID, severe combined immunodeficiency syndrome, is due to the defective adenosine deaminase gene. So gene is malfunctioning. When adenosine deaminase gene, ADA gene, is malfunctioning, that causes production of abnormal adenosine deaminase or deficiency of adenosine deaminase enzyme. Just remember, this particular enzyme, adenosine deaminase enzyme, is required for the proper functioning of T lymphocytes. T lymphocytes. You know the importance of T lymphocytes. T lymphocytes will support the B lymphocytes to produce antibodies. So here, adenosine deaminase enzyme is not produced because of the presence of malfunctioning ADA gene. Gene is malfunctioning, gene is defective. It results in the deficiency of adenosine deaminase enzyme. When that enzyme is absent, T lymphocyte becomes malfunctioning. When T lymphocyte becomes malfunctioning, they will not support B lymphocyte to produce antibodies. When B lymphocytes are not able to produce antibodies, there is no immunity power to the individual. So that's why this disorder is called as severe combined immunodeficiency syndrome, just like AIDS. The symptoms are exactly similar to AIDS, but the difference is AIDS is due to HIV infection, but this is due to genetic disorder, genetic defect in the gene. So this enzyme is crucial for the immune system to function. The disorder is caused due to the deletion of gene for the adenosine deaminase. So here the gene is absent or the gene is malfunction. How to cure this gene disorder then? See, as a first step towards gene therapy with respect to this particular disorder, lymphocytes from the blood of the patient are given in a culture outside the body. So first, from the patient body, the lymphocytes were isolated. T lymphocytes were isolated. Actually, defective T lymphocytes. And a functional ADA cDNA. ADA means adenosine deaminase. cDNA means complementary DNA. So this is a DNA, single stranded DNA at first, then it is converted into double stranded one. And this DNA is produced by the reverse transcription method. So the functional ADA cDNA using a retroviral vector. Retroviruses are used as vectors for the insertion of desirable genes into the animal cell. So a functional ADA cDNA were introduced into the human body cell, that is the lymphocytes, by the help of a retrovirus as a vector, and is then introduced into these lymphocytes, which are subsequently returned to the patients. I repeat, the healthy genes, functional ADA, cDNA, complementary DNA, were introduced into the retroviruses, and retroviruses are used as vectors. So such retroviruses were allowed to infect the T lymphocytes. Infection means here just the delivery of that functional ADA gene takes place into the animal cell. So such lymphocytes are the treated or repaired T lymphocytes, and such treated or repaired T lymphocytes were then introduced into the patient body, that means into the bone marrow. However, as these cells are not immortal means they will die. The patient requires periodic infusion of such genetically engineered lymphocytes. See, the treated lymphocytes were introduced into the bone marrow of the patient. Such lymphocytes will survive for some duration, some days. And during those days, they will provide immunity power to the body by supporting the B lymphocyte to produce antibodies. But they have certain lifespan. After that, such cells will die. Again, the patient should be provided with treated or genetically engineered lymphocytes. So, continuous introduction of treated lymphocytes is required in this case. However, if the gene isolate from marrow cells producing ADA is introduced into the cells at early embryonic stages, it could be a permanent cure. So based on this, there are two types of gene therapy. One is germline gene therapy, another one is somatic cell gene therapy. Somatic cell gene therapy is restricted to that particular patient. But if this treatment is done in the embryonic stage, the newly developed individual will contain the newly introduced healthy gene in all the body cells. So definitely it will be the permanent cure that to that newborn individual. In case of somatic cell gene therapy, so what I explained in the previous in the slide, that is a patient 4-year-old child. In such child, the treated cells were introduced into her bone marrow. So that treatment is restricted to only to that particular individual, only to that particular patient. That is not inheritable. But the same treatment is given to the embryonic stage. That individual becomes healthy and that will be transmitted even to the next generation. So this is about the two types of gene therapy, somatic cell gene therapy as well as germline gene therapy. Well, that explanation or classification is not given in our textbook. This is just for your information. So just remember these points regarding the gene therapy. Okay, the next one, application of biotechnology in the field of medicine, that is molecular diagnosis. As I told earlier, diagnosis is nothing but detection of cause for the disease. Molecular diagnosis means it is a very advanced method, a very accurate one. You know, if you go to the doctor, he will check the eyes or the mouth, the tongue. Actually, he is searching for the symptoms. You know, each disease is characterized by a specific symptom. Each disease is characterized by a set of symptoms or a specific symptom. So just by observing the symptoms, it is possible to diagnose the disease, the cause for the disease. And then he will give the treatment, doctor will give the treatment. Sometimes it becomes very difficult to identify the cause for the disease or the type of the disease just by observing the morphological features. In such case, he will advise to test the blood or the urine, or he will advise for the urine test or the blood test. When the blood or the urine is tested, it is possible to identify the pathogens in the blood or in the urine sample, if present, if the disease is very severe. In such case, the pathogens or some related chemicals, pathogenic substances or the proteinaceous components or the pathogenic products will appear in the blood or in the urine. And later, the disease will be identified and the required treatment will be given. So various such kind of diagnostic methodologies were developed, and most of such diagnostic methods are the contributions of biotechnology. Some very highly advanced methods are mentioned here for effective treatment of a disease. Early diagnosis and understanding its physiology is very important. Using conventional methods of diagnosis, that is serum and urine analysis, etc., early detection is sometimes not possible. So in such case, the recombinant DNA technology, polymerase chain reaction, that is PCR, and enzyme-linked immunosorbent assay, commonly known as ELISA, are the some of the techniques that serve the purpose of early diagnosis. So these are the advanced technology. The diseases which are not diagnosed by the serum test or the urine analysis can be detected by using these methods: recombinant DNA technology, or the polymerase chain reaction, or ELISA. Now, let me explain the PCR. Already we have studied about the polymerase chain reaction, the functioning of this PCR in the previous chapter. And here just application is mentioned. It's a very low concentration of a bacteria or virus at a time when the symptoms of the disease are not yet visible. Even in such case, the very low concentration of bacteria or viruses can be detected by the amplification of their nucleic acid by using PCR. PCR is now routinely used to detect the HIV in suspected AIDS patients. So PCR is now even used for the detection of HIV in the suspected AIDS patient. So it is being used to detect the mutations in genes in a suspected cancer patients also. You know, most of the cases, cancer is a kind of genetic disorder. It may occur because of the viral infection, but finally that causes defect in the genetic regulation mechanism. When the gene is not able to control its replication, or when the DNA is not able to control its replication mechanism, the uncontrolled DNA replication leads to the uncontrolled cell division. That leads to the development of a tumorous growth. That is called as cancerous growth or the cancer, uncontrolled cell division. So it is basically controlled by the gene itself, the DNA itself. So here, such kind of mutations can be detected by the PCR. It is a powerful technique to identify many other genetic disorders also. A single-stranded DNA or RNA tagged with a radioactive molecule probe is allowed to hybridize to its complementary DNA in a clone of the cells, followed by detection using autoradiography. So this is the step involved in DNA profiling or the DNA fingerprinting technology. So that is explained in another chapter, that is molecular base of phenotypes. Okay, so that chapter is the base for this particular chapter, for biotechnology chapter. Anyhow, the autoradiography is a step involved in DNA profiling or DNA fingerprinting technology. So by this, it is possible to detect some of the genetic disorders or the genetic defects found in a patient. The clone having the mutated gene will hence not appear in a photographic film because the probe will not have complementarity with the mutated gene. So the normal individual or the healthy individual will not contain the mutated genes, but the patient contains mutated genes. So it can be detected by the help of this autoradiography. So this is the importance of PCR for the detection of genetic disorders. Now the another one, ELISA, enzyme linked immunosorbent assay. So it is based on the principle of antigen-antibody interaction. Antigen-antibody interaction. So infection by a pathogen can be detected by the presence of antigens. So when there is an infection by a particular pathogen in the patient body, the immune system will produce antibodies against that particular antigen. So such antibodies will be found in higher volume in the patient body. So when there is a infection by a particular pathogen, specific antibodies will be produced. So in this technology, artificially synthesized antibodies are used to detect the pathogen found in the or antigen found in the sample isolated from the blood sample or the sample isolated from the patient. If the pathogen that is considered as antigen in immunology, if that pathogen or antigen is found in the blood sample of the patient, suspected patient, that will combine or that will interact with the ready-made antibodies introduced into the ELISA. So that leads to the formation of an antigen-antibody complex. So by this, we can detect the presence of antigens in the sample of that particular suspected patient, and the specific disease will be identified. So this is regarding the ELISA. It is a kind of a diagnostic method used in pathology, and these are the applications of biotechnology in the field of medicine. Okay, let me stop here. In the next video, I will continue the remaining topics of this particular chapter. Thank you.